Rahim. Welcome back, guys. Welcome to another video sessions from Kame Biology. In this video lecture we will be talking about the replication schemes of the RNA viruses. Okay, so now let me write. We talked about the replications of DNA viruses, okay? Means single-stranded and double-stranded DNA viruses. We already talked about this. But now in this video we will talk about the replications of RNA viruses. Now in replications of RNA viruses, what we will discuss? We will discuss some important terms. We will discuss the single-stranded RNA replication, we will talk in single-stranded RNA replication we will mainly talk about the positive and negative single-stranded RNA replication. And then we will talk about the double-stranded RNA virus replication. So in this topic we will talk about all these replication schemes. Okay, first we will talk about the single-stranded RNA replication viruses replications. Okay, now in single-stranded RNA virus replication we will talk about the positive and negative. Okay, first, what is positive and negative means, that what is positive single-stranded RNA and what is negative single-stranded mRNA. Now if let's suppose if the single-stranded RNA will produce the protein RNA, when the single-stranded RNA, when it use a polymerase enzyme, and with this polymerase enzyme if they produce directly the protein, they produce the protein as a product, so we call that positive single-stranded RNA. Okay, so we call that positive single-stranded RNA. When the RNA directly produce the protein component, so we call that positive single-stranded RNA. But when the single-stranded RNA first produce the positive RNA and then they produce the protein component, then we call that negative. Then we call that negative single-stranded RNA. Okay, so what we study, positive single-stranded RNA directly produce protein, and negative single-stranded RNA first this RNA is used as a template, and after that when they used as a template then they produce positive RNA, positive single-stranded RNA, and after that they produce the protein component. Means they produce protein indirectly and they produce the protein directly. Positive produce the protein directly while negative produce the protein indirectly. Fine. Now this is about the schemes of the now this is the idea about the positive and single-stranded. Let's talk about the single-stranded RNA replications. Okay, that how the single-stranded RNA viruses are replicated inside the host cell. Let's talk. Suppose this is a virus, let's suppose, having the capsid. This green one represents the capsid. Okay, now inside this virus they carry the RNA and this is positive. Okay, positive RNA. So they will present outside the cell and surrounding outside the cell, and once they reach to the host cell receptor and attach their self to the host cell receptor, so what they do, they engulf inside the host cell. Once they engulf inside the host cell, so they release their RNA. So they release their positive mRNA. As I told you that mRNA directly produces the protein, but in this case the mRNA, if the virus release the mRNA inside the host cell, in this case they not produce directly protein. What happen in this case? In this case they directly produce the negative mRNA. So what we get? We get negative mRNA. Why we get negative mRNA and why we not get the positive, why we not get the protein component from the positive mRNA? I will discuss it further, but just focus here. Once they produce the negative mRNA, then the negative mRNA again, this is the positive mRNA used as a template to produce negative mRNA. Now negative used as a template, okay? So now it is used as a template. And they directly produce what? Negative mRNA. Then negative mRNA is used as a template. Again, template. And once they are used as a template, so what they produce? They again produce, but this time they again produce more and more copy of what? So we get more and more copy of positive mRNA. These all are the positive mRNA by using negative mRNA as a template. Now after that what we get? These negative mRNA are further release what? These mRNA further release the protein, means the product. So we get the product, and after that the product means the protein, viral structure protein. What we get? Viral structural protein. So we get viral structure protein, and after that what we get? Finally we get a progeny virus. So finally we get a progeny, means this protein are used as a capsid. They are around on the positive mRNA. So this is mRNA, positive mRNA. So now the structure protein around to the positive mRNA as a capsid, and finally we get a progeny. Finally we get a progeny virus. Progeny positive single-stranded RNA virus. Okay, so that's a simple talking about the positive mRNA. What happened? Let me again explain. The positive mRNA are present around the host cell, and once they reach to the host cell, attack on the receptor, now they will engulf and they release the positive mRNA. The positive mRNA, as I told to you, that they directly produce protein, but in this case they produce the negative mRNA. Why they not produce directly protein? Because if they produce directly protein, then there will be no chances of the more and more copies of the positive mRNA. Why we need that? We need that for the copy of the viruses, for the copy of progeny viruses. If these mRNA are released in a large number, so then we will get the virus. If these mRNA are not produced more, so how we will get the progeny virus? So that's why this mRNA is used as a template for the production of negative mRNA. Once the negative mRNA are produced, then this negative mRNA is used as a template for the production of more and more positive mRNA. That's why they not produce directly the protein. First they produce this negative, and after that they produce positive, and then the positive will produce structural viral protein, and then they will around to the positive mRNA strand and make a capsid. Finally we get the progeny positive single-stranded RNA. So that's about the positive mRNA virus replication. Second, we have the negative sense virus replication. Second, we have a negative mRNA. Again we have a virus. Again, we have a virus. The virus will carry this time the negative mRNA. The virus now carry the negative RNA, negative single-strand RNA now the virus carry. Once they reach to the host cell and attach to a host cell receptor, then they released their negative single-stranded RNA. And this negative single-stranded RNA is used as a template. As I discussed to you that this negative mRNA is used as a template. Virus used negative strand. Now once this virus attached to the host cell, okay, so they will release their negative single-stranded RNA. Now this negative single-stranded RNA will release what? This negative single-stranded RNA is used as a template for the production of positive single-stranded RNA. So now it is used as a template again. As a template. So once it is used as a template, so what we get? We get positive single-stranded RNA. So once we get positive single-stranded RNA, so they not only, so this positive single-stranded RNA will produce what? They will produce the, means this positive single-stranded mRNA will produce two things. What? First they produce the product, means protein component. First they produce the product, means protein, viral protein structure. Once they produce viral protein structure, after that what they produce? They produce more and more copy of a negative stranded RNA, because we get these things. So these are all negative single-stranded RNA, and these are the viral structural protein. And finally what we get? We get a mature progeny virus. So after that what we get? We get a progeny. The capsid will around. There is first we have a negative strand, and after the negative strand will present, so this negative strand will surround, so this negative strand will be surrounded by a positive, there will be a capsid surround to the, means capsid protein surround to the negative strand. So again we get the progeny virus. We get a progeny single-stranded RNA virus carrying the negative RNA. So that's in a sense about the negative single-stranded RNA and their replications. Fine. Now last we have the, once again explain me for your understanding that what happened. This negative single-stranded RNA virus are present on the surrounding of the host cell and is present in the environment. Once they reach to the host cell, attached to the host cell receptor, so they will engulf, and once they will engulf, they release their negative single-stranded RNA. Once they release their negative single-stranded RNA, then negative single-stranded RNA is used as a template, and they produce the positive single-stranded RNA. Now this positive single-stranded RNA is used as a template to produce more and more negative single-stranded RNA, and also this positive also used to produce viral protein structure. After the production of both these two products, we get a progeny virus. So we get the negative and negative stranded RNA, single-stranded RNA will surround the capsid, and finally we get a progeny virus. This will put a pressure on the host cell and by which the host cell will burst and release more negative single-stranded RNA. Okay, last we have the double-stranded RNA virus replication. How the double-stranded RNA virus replication will occur? Once you get the idea about any one of the replication scheme, idea, so the rest of the replication you will easily understand. So anyone, if you understand the positive, if you understand the double-stranded DNA, if you understand the single-stranded DNA virus replication, if you understand any one of them a replication scheme, so the rest of the replication you will get easily. Okay, so just like in this case, once you get this idea, so this idea will really easily for your understanding. Last we have the double-stranded. Now it is very easily for you to understand because you know both of them. So if you know both of them, so the double-stranded will easily, so you will get the idea about the double-stranded DNA easily. Now how? Again we have a virus. Okay, this virus carry what? This virus carry, let's suppose this blue one present the negative, this red one present the positive. Here one question arises in our mind that the double-stranded RNA, if we talked about the double-stranded RNA replication viruses, then what kind of RNA that is? Positive or negative? Positive double-stranded RNA or negative? Now, this is both positive and negative. Why? Why this is not positive and why this is not negative? The reason behind that positive will repel each other and negative will also repel each other. So we need opposite charges. So we need negative and positive. Once we need negative and positive, so they will twist around to each other, and by twisting around to each other they will make a double-stranded RNA. Okay, so this is a double-stranded RNA. Double-stranded RNA virus. So this is double-stranded RNA virus. So we get double-stranded RNA virus. Now this double-stranded RNA virus again surround to the host cell, and once they reach to the host cell, they will attach their self to the receptor of the host cell, and what happen after that? They release their genetic material. Means they release their double-stranded DNA. Now this double-stranded DNA will release stranded RNA will release positive mRNA by using RNA polymerase. So they use RNA polymerase enzyme. Once they use RNA polymerase enzyme, so after that what happened? They will release positive mRNA. So what we get? We get positive mRNA. This blue one is positive mRNA. So we get positive mRNA. With this positive mRNA, we get two things. Now this positive mRNA is used as a template for the production of negative mRNA. Single-stranded RNA, okay? And also produce the protein structure, viral protein structure. So also release the viral protein structure, and after that, what happened? After that, this negative single-stranded RNA is used as a template for the production of more and more copy of negative as well as positive. So we get negative and we get positive. We get negative and positive RNA. What we get? We get negative and positive single-stranded RNA. Fine. What we see in this double-stranded RNA replica, first they release the double-stranded RNA as a genetic material. Once they release double-stranded RNA as a genetic material, after that they release the positive mRNA, single-stranded. Okay, so once they produce positive single-stranded RNA, after that they produce the negative single-stranded RNA. Now this positive single-stranded RNA is used as a template, not only produce the negative mRNA, they also produce the viral protein structure as well. Viral protein structure. After that, this negative mRNA is used as a template for the production of more and more copy of positive as well as negative mRNA. Okay, so red is negative and blue is positive. So we get more and more positive and negative mRNA. After that, what we get, as both are opposite charges to each other, so we get just like this form. So we get the DNA in this form. RNA. So we get RNA in this form. So now this is double-stranded RNA. And finally, this protein will surround and make a capsid. Why we call it progeny? It is the copy. Progeny means copy. Progeny double-stranded RNA virus. We get the progeny double-stranded RNA virus. So that's in a sense about the replication scheme of the RNA viruses. I hope this video helpful. If you like the video, please set the like button, share the video and subscribe the channel. Okay, don't forget this. Subscribe the channel to get more interesting video lecture like that. Thank you.